Good day for everyone. I'm Luis Guerra and I want to introduce you the results of my doctoral thesis in marine sciences, seagrass as carbon sink, an assessment applied to Colombian Caribbean. This thesis was advised by Jose Mancera from National University and by Martin Zimmer from San Andrés from Deutschland. Okay, I want to explain you the concept of blue carbon. Well, blue carbon is all the organic carbon that you can find in the marine and coastal ecosystems across the world. In this case, I conduct an study in the southwestern Caribbean, specifically in seagrass meadows. As you can see, for Cohen and others, conduct a study in the 2012 where the global blue carbon in seagrasses was studied, and as you can see, there are few data in the southwestern Caribbean in the tropical Western Atlantic region proposed by Furcuron. In this case, he did this analysis using just the seagrass living biomass of many of the species of seagrass plants in these places. Also, he conducted an analysis in the sediment using an equation based on the organic matter content in these sediments and the total organic carbon or TOC percentage in these sediments. Also, in this study was presented a high variation in means between all the regions because there are many particular features of these ecosystems. Many of these features are physical chemicals, and this variation must be studied specifically inside each region. In this case, I conduct my seagrass blue carbon assessment in the study area located in the Reef Lagoon in the San Andrés island, part of the archipelago of San Andrés, Providence, and Quetlina, located in the southwestern Caribbean and under the political jurisdiction of Colombia, but close to Nicaragua. As you can see in this map, there are 4 coverage of seagrass meadows. All of these meadows are inside a coral reef lagoon and protected by a coral reef barrier. Also, close to the littoral, you can see some coverage of mangrove. Also, the proposal of my thesis was to assess the stock of organic carbon in these seagrass meadows, but also I use one of many of the processes linked to the budget of blue carbon, like the case of above ground growth, to explain what is happening here. Also, I use the physical features of this system to explain some variabilities in the means of organic carbon and seagrass living biomass here in the study area. In this thesis, I conduct an analysis in sediments below the seagrass meadows and also in the seagrass living biomass, classifying this biomass like the whole biomass of all species in these meadows, and specifically the case of Thalassia testudinum. I want to start explaining you the results of the first assessment in sediments conducted in this study area. As you can see, I use 2 equations to estimate the organic carbon stock in this area. In San Andrés, using the equation from Fourcuron based on the organic matter, and also the equation established here in this study using a regression analysis and a linear relationship found among the organic matter in sediments and organic carbon in these sediments. As you can see, the mean using these two estimators suggests the mean of the study area, I mean the organic carbon in this area, is comparable or similar to the South Atlantic and tropical weather Atlantic mean. In another hand, I assess the seagrass living biomass using the whole data collected from the study area. As you can see, the mean of organic carbon stock in San Andrés Island is similar to the global mean. In this case, I present you Thalassia testudinum presented the 78% of the seagrass living biomass of whole meadows. Okay, but the question is, these 72 replicates or sampling points were coming from? Data come from a sampling design considering some variations in the weather seasons or sampling periods, and also variations linked to the exposition or shelter conditions of some sampling sites within the reef lagoon, and also some sampling points inside each sampling site where I suggest the name of depth stations. In this case, I am linking these possible variations to the light and turbulence factor. I want to explain my sampling design. As you can see in the map, I must to make a cartography of the seagrass meadows present in the island coast. As you can see, you see here the coral reef barrier in the east of the island. As you can see, the island of San Andrés in the east side. In the middle, there are seagrass meadows, and I have to verify the presence of these seagrass meadows using some criteria that I established here. Specifically, meadows where the coverage of seagrass is over 30%, and also patches higher than 1 hectare. The result was a map of these seagrass meadows where the seagrass within the reef lagoon have 314 hectares. Also, I found mixed meadows where you could find Thalassia testudinum, Syringodium filiforme, and Halodule wrighti. The dominant species here is Thalassia testudinum across all the sampling design. Here in the map, you can see the SPB site or sprat bite sampling site is located in the north section of the reef lagoon, where there are a gap in the coral reef barrier So I expect exposed conditions linked to some physical factors affecting the stock of organic carbon here in this site. But in the middle section, we can find two more sampling sites. MOD, or the harbor, or OPR, Old Point Regional Mangrove Park, in a sheltered condition, protected from the effects of winds and waves coming from the northeast. Also, it's possible to find some effects of the runoff from the littoral because the island is mainly flat, but many of the runoff runs to the littoral in the rainy season. Also, it's possible to assess the seagrass meadows in the southern section of the reef lagoon, where there are a big gap where also exposed condition is possible to be assessed. Also, I repeat my sampling design in 2 points or 2 times or sampling periods in the year. This data in this graph show you the means and standard deviations of the precipitation of the last 5 years. As you can see, it's possible to find a dry season starting the year and have a rainy season in the end of the year. Conditions of the study area are really interesting because we are located under a microtidal regime and also really far away from the effects of the continental runoff. Then it's possible to suppose the organic carbon produced here in this big system, in this coral reef system, is alloctonous. I mean, the organic carbon could be produced locally here in the system, within the system. Also, as you can see in the graph, in the sketch, seagrass meadows within the reef lagoon are located in the slope close to the littoral of the island. I mean the western section of the reef lagoon. Then it's possible to assess at least 3 intervals of depth where the deep water is located far away from the coast, middle water in the mid, and shallow water are really close to the shoreline. Before to continue with my results, I have to explain something. Within each sampling site, I established 3 depth stations, 3 intervals of depth where I collect data to increase the representativeness of my sampling within each sampling site. These depth stations are coded like this. For example, sampling site with the number 1 corresponds to deep water sampling interval, with the number 2 mid-water sampling station, and with the number 3 shallow water sampling station, all within the same sampling site. Okay. Respect to my results in sediments, to explain the effects of the factor waves intensity, we can see here in my results there are significant differences among the conditions of the shelter sites respecting to exposed sites. If you see the figure in the right hand above, you see MUD and OPR, the shelter sampling sites, presents a mean higher than the exposed sampling sites. What it means? The exposed sampling sites have a high percentage of sediments with size higher than 63 micrometers. Also, I want to explain this is the results of the effects of the high hydrodynamic flux in these conditions of exposed sampling sites respecting to the shelter conditions observed in these shelter sampling sites. Also, I take all the categories of size of sediments using my granulometric analysis to construct an index that I can use like a resume of the profile of granulometry of these sediments in the study area. I use the Shannon-Wiener evenness index. As you can see, higher evenness index are linked to a higher evenness, but low evenness index are linked to some size of sediments dominant over the other ones. As you can see, the evenness profile in the shelter sampling size are really different from the non-evenness profile in the exposed sampling sites, where sandy sediments are dominant. Also, using the same categories of granulometry, like size of sediments, it's possible to conduct an analysis of coordinates or principal coordinates using Euclidean distance. As you can see, SPB3 and RCB3, both sampling sites in exposed conditions and also in shallow water conditions, are really different or have high distance or dissimilarities respecting to the other sampling sites and sampling stations. Also, there was significant differences among other variables in sediments, like the case of the organic carbon. Total organic carbon in these sampling sites was different among the samplings in shelter conditions, and it was lower in the exposed conditions. In this case, I present the results using two equations. The equations proposed by Fourcuron in their global assessment, and what I found in this study using talc and organic matter percentage in the sediments collected in the study area. Okay, as you can see, organic matter could be used to estimate the organic carbon stock in sediments. In this case, I'm presenting you the data of the organic matter in sediments. using an analysis to compare the stock of this organic matter among sampling sites, but also among depth stations within these sampling sites. As you can see, the means in SPB and RCB are lower than in the case of MUD. Remember, SPB and RCB are in exposed conditions. Also, you can see here the mean in the shallow water interval presents lower values than the deep water interval of the sampling site. The same case is happening in SPB. Then it's possible to propose a relationship among the organic matter and the size of the sediments. If you have sites where the percentage of mud-like sediments is higher, then it's possible to find also higher values of organic matter and consequently the mean on organic carbon stock. Okay, but what I observed in sediments are similar respecting to I found in the seagrass living biomass. I mean, the same effects linked to the conditions of sheltered or exposed sites in sediments could be observed in the seagrass living biomass. The same pattern? Well, the first difference is I found significant differences among the seagrass living biomass among dry season respecting to rainy season. Higher values or means was observed in the dry season or sampling period. These differences was not observed using the sediments variables. Then there are effects of the seasonality in this study area. Then I propose to analyze what is happening with the data from the rainy season and the data of dry season. Also, as you can see in this graph, you have the data of the whole biomass of seagrasses. I mean Thalassia, Halodule, and Syringodium. But in this other graph, we have only the data from the Thalassia testudinum, because Thalassia testudinum is the dominant species. As you can see, the same pattern or same differences was observed in the whole biomass of seagrasses and also in the exclusive biomass of Thalassia testudinum. Well, but the seagrass living biomass could be divided or split in two groups or two components. The above ground component, like green leaves, and the below ground component, like roots and rhizomes biomass. It's possible to find some differences among these components. Well, what I found is the below ground biomass is really higher respecting to the above ground biomass of the whole species in these seagrass meadows. And also there are differences linked to seasonality among these samplings periods, where biomass is always higher in dry season respecting to rainy season. Okay, there are some differences among sampling periods. But what is happening with the effects of the coral reef barrier effect on the mitigation of the wave intensity in these two kinds of sites, I mean exposed and sheltered sites? What I found is MUD have higher values than the others in the in the rainy season. And also, if you consider exclusively the biomass, the living biomass of Thalassian testudinum, we can find a similar pattern where the shelter sites present higher and significant values respecting to exposed sites like RCB and SPB. A similar pattern was observed in the dry season. The difference among the dry season and rainy season is just the magnitude of the seagrass living biomass stock. Well, in this slide, I want to show you something. If you consider variables like organic matter in sediments as percentage in the left hand and in the right hand, you see the percentage of mud-like sediments in these sampling sites. It's possible to find a qualitative relationship among these three variables. Where you find seagrass living biomass with higher values or higher means, also it's possible to find high percentage of organic matter in sediments and also higher percentage of mud-like sediments. Well, but these variables like organic matter in sediments or mud-like sediments are related to the seagrass living biomass? Well, my answer is yes. There are a relationship, but we have to understand what is happening with the components in this seagrass species, specifically in the case of Thalassia testudinum, the dominant species of these meadows. Well, to answer the last question, we have to consider something. Seagrass living biomass could be divided in components according to the method. If you use the method of loss on ignition to determine the organic matter in this biomass, it's possible to obtain the organic matter and also the inorganic matter fraction of this biomass. But using the autoanalyzer method, it's possible to determine some components, some chemical components of this biomass, like total organic carbon or total nitrogen. In the case of the work of Duarte, he proposed the biomass of all seagrasses have an average of 34% of this seagrass living biomass as organic carbon. Then Furcuron proposed this coefficient to estimate the organic carbon stock in seagrass living biomass, just multiplying this number by the seagrass living biomass. I conducted an analysis and I found significant differences among the organic carbon stock in green leaves respecting to below-ground modules or organs in seagrass, specifically in Thalassia testudinum, where roots and rhizomes present similar means. The mean of organic carbon in these samples was similar to 34% proposed by Duarte, but the difference among these two analyses is Duarte used just green leaves in his analysis. Well, but also I found some significant differences among the percentage of total nitrogen in green leaves and roots respecting to rhizomes in Thalassia testudinum. Then it's possible to suggest roots is used by Thalassia testudinum or are used by Thalassia testudinum to collect and to sink nitrogen in its biomass. Something different from the case of carbon, of organic carbon, where roots and rhizomes present similar percentage among them and different from green leaves. Also, you have to remember, in average, above ground biomass, I mean green leaves, have 5 to 7 times more organic carbon than below ground biomass. Then it's possible to suggest a relationship among the nitrogen, organic matter, and roots in these meadows, because typically Caribbean Sea presents limitations by nitrogen respecting to phosphorus and other nutrients. Then organic matter could be a source of this nitrogen, and this nitrogen could be obtaken by the roots, and that assumption could be approached if you consider the ratio among these organs or modules in seagrasses. And in this case, I show you this table where the shelter sites like MUD and OPR present higher roots to green leaves ratios than the exposed sites. Then it's possible to suggest in shelter sites, seagrass meadows invest more of their biomass in roots than in green leaves, different from the case of exposed sites. If you compare the ratio among rhizomes and roots, it's possible to observe higher ratios in exposed sites than the ratios observed in sheltered sites. It means exposed sites are investing more of their biomass to conserve rhizomes, to create rhizomes. Then it's possible these rhizomes are a strategy to collect or to reserve nutrients, but also to tolerate the effects of the hydrodynamics linked to exposed conditions within a coral reef lagoon. And in the case of shelter shelter sites, it's possible seagrass meadows there are using their roots like the media to obtain nutrients available in these rich soils or these rich sediments respecting to the content or the stock of organic matter deposited there. Also, I tried something new. whole biomass of Thalassia testudinum, for example, could be divided in two portions using the loss of ignition method, like the inorganic biomass or ashes and the organic biomass or volatile matter. When I determine the organic matter stock in this seagrass biomass, I found not significant differences among stocks in these sampling sites. But if I consider just the percentage of organic matter in this sampling site, significant differences was observed. But what is explaining these differences among sampling sites? Well, these differences are linked to the capacity of the below-ground modules or organs to reserve inorganic materials like minerals. For example, if you see in the left figure, we have data of green leaves in all sampling sites, but there are not significant differences. But in the right figure, you can see significant differences among sampling sites. In this case, MUD present the lowest value of organic matter in Thalassia testudinum biomass. Then it's possible to suggest some sampling sites use the biomass to store some minerals or other inorganic components. Okay, what is happening with the organic carbon stock within each sampling site? I mean, the effects of the depth or the distance in this seagrass meadows organic carbon stock. As you can see, we have here data from the whole biomass of all seagrasses in these meadows and just the biomass in Thalassia testudinum in the right figure. As you see the circles are marking the exposed sampling sites. As you can see, the sheltered depth stations present higher seagrass living biomass than the deep water sampling stations. These differences are possible because in shallow water, seagrasses have to develop higher biomasses to conserve the stability in this high turbulence environment close to the shoreline, where the waves have the higher effects on the seagrass meadows. And as you can see, these sampling sites used to produce more rhizomes than roots. In this case, these meadows are investing biomass in rhizomes to increase the stability in this turbulent environment. The same pattern was observed in the case of the dry season data or sampling period in 2017. Then, whole living biomass in seagrass meadows within a reef lagoon is affected by some biotic factors and processes. As you can see before, there are environmental factors affecting the organic carbon stock according to the place and according to the depth. But there are some biotic factors or processes that could be affecting this seagrass living biomass and their organic carbon stock? Well, I tried with these factors. I found not similar patterns among seagrass living biomass in Thalassia testudinum and these variables like leaf growth or the turnover rate, the average of leaves per shoot, leaf length or width, senescent biomass or brown biomass, epiphytic organic biomass, or the incidence of labyrinthula, like a pathogen of Thalassia testudinum. As you can see in this figure, I am presenting the living biomass in Thalassia testudinum and the difference observed among the sampling sites. I expect to find similar patterns assessing these variables. This pattern was only observed using the shoot density variable in Thalassia testudinum. And also, a similar pattern was observed using the dry biomass of macroalgae and other senescent material linked to these seagrass meadows. Then it's possible to suggest shelter sites present higher values in mud-like sediments, in organic matter, in seagrass living biomass, in shoot density, and also in macroalgae biomass, usually linked to some genus like Alimeda and Opuntia, coralline macroalgae. Also, I found a relationship among the organic matter in sediments and the living biomass in the below ground component in Thalassia testudinum. It supports the hypothesis that below ground modules or organs is used to uptake these nutrients linked to the organic matter in sediments. Conclusions. Organic carbon stock in the living biomass of seagrass in San Andrés Island is comparable with the Western Atlantic region, usually lower than the global mean. Organic carbon in sediments was higher than the Western Atlantic region, and it was similar and close to the global mean. Biomass is not the better estimator to calculate organic carbon stock in seagrass meadows within a reef lagoon, because we have to consider the organic matter or the organic carbon in this biomass, and this biomass could be compressed also by inorganic fractions. But the use of the coefficients to estimate organic carbon from living biomass could be appropriated. Inorganic matter stock in seagrass biomass vary according to the wave's intensity. Then there are effects of regulation of this coral reef barrier on the process or function of accumulation of organic carbon in the meadows of these seagrasses. Shelter sites presented higher seagrass living biomass than exposed sites, and this is responding the hypothesis that coral reef barrier is enhancing the capacity of organic carbon accumulation in these seagrass meadows. Organic matter in sediments could be a nutrient source in seagrass meadows in environments limited by nitrogen. Organic matter in sediments was related to below ground living biomass in seagrass meadows. Most of the organic matter and organic carbon within seagrass meadows is autochthonous, but there are contributions linked to the contiguous organic carbon source, like mangroves or coral reefs. Exposed seagrass meadows presented variations in the textural attributes of the sediment, which allows deeper meadows to have higher mud-like sediments percentage in contrast to the shallower ones near to the shoreline. I have some recommendations here related principally with new studies and also suggesting some changes in the monitoring program conducted by the environmental authority in San Andrés, because the current studies and current monitoring program is using variables linked to the above ground environment in the seagrass meadows, and it's possible to have not enough information to assess the integrity of this ecosystem if we are not considering the conditions of the below ground component of the below ground pool in these ecosystems. Thank you for all everybody that gave me a little help to conduct this study, especially to the box in set empty. Thank you so much.